Hello everyone. I am Dr. Bhavna Mattu from Department of Physiology at AIIMS, New Delhi, and I will be covering module number 4 on force, mechanics and muscle metabolism from the subject of physiological biophysics. Today our learning objectives are to understand the mechanical model of muscle, to define a motor unit, to know the types of muscle fibers, differentiate between the types of muscle contraction, and finally to understand muscle metabolism. This module will be covered under the heads of mechanical model of muscle, motor unit, fast twitch and slow twitch fibers, types of muscle contractions, factors controlling force generation by muscle, and muscle metabolism. Now, force is a push or a pull on an object causing its displacement, while mechanics is concerned with the behavior of physical bodies when they are subjected to such force. In the human body, muscle is the basis of generation of any force, and the skeletal muscles control body's voluntary movements. It is thus imperative that we understand the force generation by the muscular system and the mechanics behind it by using simple concepts of elasticity, length, tension, velocity and force. We will also attempt to understand the fuel required and the effect of different energy substrates on actual work done. Learning of such concepts has important applications, especially in the fields of sports and exercise physiology. Now, the three-component model is used to explain the mechanics of skeletal muscle contraction. You can see in this figure, it consists of three elements: the contractile element, the series elastic element and the parallel elastic element. Contractile elements seen in red consist of thick and thin filaments that interact through cross-bridge cycling and generate force or active tension, while elastic elements contribute to the development of passive tension in the muscle when they are stretched. Elastic elements are classified into two types based on their pattern of arrangement with respect to the contractile elements, as parallel elastic elements, which consists of the sarcolemmal and the connective tissue components disposed parallelly along the muscle fibers, and the series elastic elements. Now, these consist of tendons that are serially connected to the contractile elements of the muscle fiber. Every muscle is supplied by a motor nerve that electrically excites the muscle fibers to produce a contraction. A motor nerve consists of a bundle of motor nerve fibers, which are the axons of the alpha motor neurons located in the anterior column of the spinal cord. Every axon, when it reaches close to the zone of innervation, divides and forms multiple axonal branches that form terminal boutons and synapse with the muscle fibers. As you can see in this figure, the alpha motor neuron along with the set of muscle fibers innervated by its axon is collectively called a motor unit. The fibers constituting a specific motor unit could be spatially scattered within the muscle belly, and with every action potential fired by the alpha motor neuron, the muscle fibers forming the corresponding motor unit contract in an all or none fashion, which means when the impulse arrives at the motor unit, it will either contract or it won't. This is all or none. There is no gradation in the response of a motor unit. But does that mean all the muscle fibers are similar? No, that is not the case, and muscle fibers can be classified into two. This is based on the time it takes to develop peak tension when they contract. Muscle fibers that develop peak tension in a very short duration of time falling less than 10 milliseconds are classified as fast twitch fibers, while the ones taking 40 to 100 milliseconds are labeled as slow twitch fibers. Slow twitch and fast twitch muscle fibers also differ in their metabolic capacity to sustain aerobic and anaerobic pathways. The difference in the rate of tension development has been attributed to the different isoforms of myosin expressed in the muscle fiber that differ in their ATPase activity. Skeletal muscles also differ in the proportion of fast twitch and slow twitch muscle fibers present in them, and thus the rate of tension development in the whole muscle is influenced by this ratio. Not only are the muscle fibers inherently different, and this difference can also be in the type of the contraction. And to understand this, let us look at the first graph, which shows an in vitro preparation of an isolated muscle which is fixed at two ends, and therefore the length is constant and is shown by the straight yellow line. And after electrical stimulation, the changes in tension are observed and as seen by the blue curve. Such a contraction where the length remains constant is called isometric. Iso meaning same and metric meaning length. The second type of contraction can be appreciated in the second graph, where the tension depicted in the blue line remains constant while the length changes. Isometric contractions are observed physiologically when muscle contracts against a load exceeding the maximum total tension it can develop. Example, when you use both your arms and you try to push against a wall. That's an isometric contractions. While most of the body movements at various limbs and joints against a definite load are mostly isotonic in nature. Isotonic contractions are further subclassified into two based on the conditions of loading as preloaded or afterloaded. Preloaded contractions are in which the load is already acting on the muscle before it starts to contract. Example, when a heavy box is to be lifted and placed on a table, we let it hang from our hand first before we can finally lift it and put it on the table. Whereas in afterloaded contractions, the load starts acting on the muscle only after it shortens by a finite length, like suddenly lifting an object without letting it stretch the muscle. To understand better the mechanics of isometric contraction, the relationship between total, active and passive tensions and the length of the muscle is studied. Passive tension is exerted by the muscle when it is in the relaxed state and is contributed primarily by the series and the parallel elastic elements in the muscle when they are stretched. Active tension, on the other hand, develops in the muscle when it contracts, and this is due to actomyosin cross-bridge cycling. The total tension thus is equal to the sum of active and passive tensions. The length-tension relationship can be studied by fixing the two ends of the muscle and stretching it in a graded manner to alter the length. In the graph, you can see on the x-axis is the length of the muscle and on the y-axis is the peak tension of the isometric contraction. With the graded increase in length, active tension rises as is seen in red and peaks at the optimal length or OL and then decreases with further stretching of the muscle. Passive tension seen in blue rises once the muscle is stretched beyond its resting length or L, and after which it increases exponentially when the muscle is stretched further. When the muscle is detached from all its bony attachments, it attains a shorter length. This is known as the resting length or L. Stretching of the muscle in a graded manner initially leads to the increase in active tension generated, which peaks at around 1.2 times the resting length of the muscle. Further stretching of the muscle leads to a reduction in active tension and an exponential rise in the passive tension exerted by the elastic elements. The molecular basis of the peaking of active tension at 1.2 L has been attributed to the attainment of maximal acto-myosin interactions because of optimal overlap and increased proximity due to compression of the myofibril lattice because of stretching. Now, to assess the mechanical performance of the muscle contracting isotonically, the relationship between the load or the force of contraction and the velocity of shortening is studied, and this is expressed as the force-velocity relationship. In isotonic contraction, the force of contraction is found to be inversely related to the velocity of shortening, as shown in the figure. Maximal velocity of shortening, or Vmax, is attained when the muscle contracts against a zero load or in an externally unloaded state. As the load increases, the velocity of shortening decreases, and finally it becomes zero against a load Fmax that cannot be moved when the contraction completely becomes isometric. With this understanding of the motor unit, the types of muscle fibers and the contractions, let us look at the factors which control force generation by muscle. Force generation by muscle in vivo is determined primarily by two factors: the intrinsic contractile properties of the muscle fibers and the pattern of activation of motor units in the muscle by the nervous system. To understand this better, let's look at this figure which shows the contractile response of an isolated nerve muscle preparation to a brief electrical stimulus applied to the nerve, and this is called a simple muscle twitch. If the same preparation is stimulated repeatedly with a train of stimuli with interstimulus interval longer than the refractory period, we may observe a partial or a complete fusion of the contractions depending on the frequency of stimulation, leading to the smooth sustenance of the tension generated by the muscle, which is seen in the next figures. The state of complete fusion of contractions is known as tetanus, and this occurs when every subsequent stimulus excites the muscle to contract before it starts relaxing after the previous contraction. It is important to observe that the peak tetanic force is always greater than the peak twitch force. This has been attributed to the greater availability of calcium in the sarcoplasm when the muscle is repeatedly stimulated, as pumping back of calcium will not be complete before the next release is triggered. Additionally, fusion of contractions retains the series elastic elements in the continuously stretched state, thereby transferring entirely the work done by actomyosin cross bridges to generation of tension within the muscle. The physiological mechanism by which gradation of force of muscle contraction is ensured in vivo is through a phenomena known as motor unit recruitment. Now, nervous system activates more number of motor units when the force required to move the limb against a load increases. Activation of more number of motor units will increase the total summated force generated by the muscle. Additionally, the activation of different motor units takes place in a slightly asynchronous manner, because of which the summated force shows smooth transition due to overlap of contractions from different motor units. Motor unit contractions taking place in the body are either incomplete or complete tetanus. Now, contraction of the muscle also requires a continuous energy supply for the actomyosin cross-bridge cycling and the pumping back of calcium, which is released with every excitation back into the sarcoplasmic reticulum. and maintenance of the ionic gradients across the cell membrane. This rate of energy requirement is heavily dependent on the power output or the rate of work done by the muscle. Sources of energy substrates for the contracting muscle are the stored ATP, creatinin phosphate which combines with ADP and rephosphorylates it to ATP, and the glucose derived from the stored muscle glycogen, and finally the plasma glucose and the body fats. If we look at the temporal availability of these substrates, stored ATP can meet the energy requirements for almost a second, while creatinin phosphate can meet the energy requirements of a maximally contracting muscle for about 3 seconds. Stored muscle glycogen can provide glucose through glycogenolysis, which can meet the energy requirements of the muscle for up to minutes, while plasma glucose and body fats can sustain the contractile activity of muscle beyond a couple of minutes. In a post-absorptive state, plasma glucose is maintained primarily through the liver glycogenolysis. With increase in the duration of the muscle activity, free fatty acids mobilized from the adipose tissues undergo oxidative metabolism to supply ATP for the muscle contractions. Glucose can also be utilized to generate ATP by exclusively anaerobic or by both aerobic and anaerobic pathways. Anaerobic glycolysis drives the energy production, especially when the oxygen supply to the muscle cannot catch up with the rate of energy requirement. However, the number of molecules of ATP generated per molecule of glucose metabolized anaerobically is significantly low when compared to aerobic pathways. Exclusive usage of anaerobic pathways can sustain the energy requirements of heavy bouts of exercise lasting only for seconds, such as a 100-meter sprint. Muscle fibers differ in their ability to derive energy through these anaerobic and aerobic pathways. Slow twitch fibers have greater aerobic capacity than fast because of greater abundance of mitochondria, higher capillary density and myoglobin levels, while fast twitch fibers have higher glycolytic capacity than slow twitch fibers. Finally, to summarize, the three-component model of muscle consists of contractile elements, series elastic elements and parallel elastic element. An alpha motor neuron along with the set of muscle fibers innervated by its axon is collectively called a motor unit. And muscle fibers that develop peak tension in a very short duration of time, falling less than 10 milliseconds, are classified as fast twitch fibers, while the ones taking 40 to 100 milliseconds are labeled as slow twitch fibers. In isometric contraction, the length of the muscle remains constant while the tension developed changes, while in isotonic contraction, the tension developed by the muscle remains constant and the equivalent to the load applied, while the muscle shortens during the phase of contraction and lengthens back to its original length during the phase of relaxation. Force generated by the muscle in vivo is determined primarily by two factors: intrinsic contractile properties of the muscle fibers and the pattern of activation of the motor units in the muscle. The physiological mechanism by which gradation of force of the muscle contraction is ensured in vivo is through a phenomena called motor unit recruitment, and contracting muscle derives energy from stored ATP, creatinin phosphate, glucose derived from stored muscle glycogen and plasma glucose and body fats. Glucose further can be utilized to generate ATP by exclusively anaerobic pathways or by both aerobic and anaerobic pathways.